Alright y'all, in this video we are going to go over nucleic acids and how they're connected to each of the units that we've covered this year. The first unit of the year we talked about macromolecules. So we're going to look at the foundational knowledge that you need to know about nucleic acids from a macromolecule standpoint, starting with the chemical structure of nucleic acids. So first of all, nucleic acids are made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus. These elements are arranged to form the monomers, and the monomers are held together by phosphodiester bonds. Those monomers that are held together by the phosphodiester bonds are nucleotides. It's important that you know that nucleotides are made up of three parts. You have a phosphate group, a 5-carbon sugar, and a nitrogenous base. The nucleotides differ slightly depending on which nucleic acid we're talking about. Is it DNA, deoxyribonucleic acid, or RNA, ribonucleic acid? If you're talking about DNA, the monomer is missing one OH group. Hence the name deoxy in deoxyribonucleic acid. RNA, on the other hand, does have that OH group, therefore the name ribonucleic acid. Those OH groups I'm talking about are on the sugar part of the nucleotide. So the deoxyribo sugar is the sugar that I'm talking about without the OH group. The ribose sugar is the one that does have the OH group and that oxygen. Now, it's important to know how the structure of these nucleic acids are going to affect the function of DNA. Remember, DNA is used to store and transmit genetic information. So DNA is double-stranded. It's more stable because those nitrogenous bases are held together by hydrogen bonds, and it contains that deoxyribose sugar that I was talking about a minute ago. RNA is single-stranded, it's not as stable, and it consists of a ribose sugar. Now, we can also talk about the differences in the structure of each nucleotide because there are 5 different nitrogenous bases that could potentially be attached to a nucleotide. The order of these nucleotides and those nitrogenous bases is what eventually is going to lead to the organism's genetic makeup, what makes it the way that it is. 2 of these categories exist. We have purines and pyrimidines. Purines are the nitrogenous bases that are made up of 2 rings. Those are adenine and guanine. Pyrimidines, on the other hand, have 1 ring. Those are cytosine, thymine, and uracil. In Unit 2, we talked about cell biology, which is a great time for us to point out and remember the differences in the nucleic acids and the structure of those in both prokaryotes and eukaryotes. Prokaryotic DNA is circular, and it is made up of a single chromosome. This single circular chromosome doesn't have any histones, which are the proteins that are used in eukaryotic cells to help organize and store the DNA. Prokaryotes don't have any of these. They are instead supercooled. They do not contain any introns. Their genetic information is the way that it is. There's no introns. And because there's no nucleus, it is going to be found floating in the cytoplasm in a region that we call the nucleoid. Eukaryotic DNA is linear, and there are more than 1 chromosomes. Because this DNA is linear, and there's multiple of them, our cells need to keep these organized by using histones. These are protein complexes that interact with the DNA and the charge of the DNA and wrap up our DNA in order to keep it compacted. It does contain introns, which are regions of DNA that are going to be cut out and they're not used to code for proteins. And because there is a nucleus, the DNA is stored in the nucleus. In Unit 3, we talked about energetics, and this is a great time to go over the process of DNA replication and the type of reaction and process that it is. So, DNA replication is an endergonic process, meaning that it has a positive delta G, or a positive change in free energy. It requires ATP in order to synthesize our new DNA, and it is regulated by enzymes. Another important molecule that we talk about in the energetics unit is ATP, adenosine triphosphate. Adenosine triphosphate is a nucleic acid. This is an adenosine molecule plus 3 phosphate groups. So a regular nucleotide like you're used to seeing it has a phosphate group, a sugar, and a nitrogenous base. In this case, you have the phosphate, the sugar, and the nitrogenous base adenine. But instead of just one phosphate, you have 3 phosphates. Now, the hydrolysis of ATP, which is the breaking of that last phosphate group, is going to give us adenosine diphosphate, and it's going to release the energy that was stored in the bond between the last phosphate and the ATP. Now, ATP is used to kind of carry the energy between endergonic and exergonic reactions. I like to think of it as like energy currency for the cell. It's kind of like a debit card or bank account. It just holds on to the energy until it needs to be used. In Unit 4, when we talked about cell division and Mendelian genetics, DNA was brought up a lot. This is really where nucleic acids shine. Starting with the cell cycle, we have our DNA synthesis happens in the S phase. We are going to replicate that DNA. It's regulated, and this is where we check for mutations. Then, when the cell is ready to divide, our loose DNA in the form of chromatin is going to cool up in mitosis and form the chromosomes that you're used to seeing, those little X's. In prophase 1, we also have crossing over that happens, and this is important because it increases genetic variation by exchanging little parts of DNA on the other homologous chromosome. Another way that we increase genetic variation is through fertilization, which is where you combine the DNA of an egg and a sperm. The third way that we increase genetic variation is through independent assortment, which is where we have the chromosomes lined up in the middle randomly, which is going to alter what new gametes are being formed and what combination is in each of those new gametes. Another way that we talk about genetics or DNA in the Mendelian genetics is because we can trace genetic disorders through pedigrees, which are the family tree things. The other unit where DNA plays a major role, or nucleic acids play a major role, is in Unit 5, where we talk about molecular genetics. This is where we really got into the nitty-gritty of DNA replication, and there are a ton of enzymes that play really important roles in DNA replication. Some of those are the DNA polymerases. You have DNA polymerase 1 and 2. Those are both going to attach nitrogenous bases to the newly forming strand of DNA. You have ligase, which forms the phosphodiester bond between the phosphate groups and the sugar groups of the phosphodiester backbone on the newly synthesized strand of DNA. You have helicase, which opens up the strand of DNA by breaking the hydrogen bonds between the nitrogenous bases. And you have topoisomerase, which is used further down the strand of DNA to prevent supercoiling and to remove stress from the unwinding strand. You also have single-stranded binding proteins that are going to help to keep those opened up strands open and prevent the hydrogen bonds from reforming between the nitrogenous bases. You also have histones, which are the protein complexes that are wrapped up, or that DNA is wrapped around. The structure of DNA was discovered and worked out through a series of experiments that all led to other experiments and eventually the discovery of DNA. So there were a lot of scientists involved in this. It wasn't just one person. There was a lot of collaboration, whether intentional or unintentional. I'm throwing shade at Watson and Crick there. But yeah, that's something that just the scientific community, the scientific process is just crazy wild sometimes. It's also important to know that mutations are what change DNA. There are several different kinds. The main two categories are point mutations and frameshift mutations. A point mutation is a change in a single nitrogenous base, and a frameshift is one where you shift the reading frame all the way over one way or another. DNA technology is also an important part of the unit that we talked about. We talked about a lot of the current and upcoming DNA technology, including CRISPR, gel electrophoresis, and polymerase chain reaction. We talked about how gene expression is controlled, so ways that we can change the DNA structure in order to either increase or decrease transcription depending on the protein that we need. So we can do methylation, which is going to silence the gene, meaning the protein is not going to be made. Or we can do acetylation, which is going to increase the production of that protein. We also talked about bacterial DNA changes through transformation, transduction, conjugation, and transposons. Finally, we talked about evolution, and nucleic acids are important because DNA is, or mutations really, are the driving force of evolution. Without genetic diversity that comes from DNA, then there is no evolution. DNA can also be analyzed in order to follow evolutionary history and to make phylogenetic trees. The way that we do this is by comparing DNA samples. The more similar DNA is, the more similar the organisms are, and the closer their relationship is, evolutionarily speaking. All organisms use the same code, meaning the same amino acids are coded for by the same sequence of nitrogenous bases. This is evidence that this structure of genetic information and transmitting genetic information and inheritance was present in the last universal common ancestor. That's it for nucleic acids. Remember, if you have any questions, feel free to shoot an email to your teacher. Good luck!